Thank you, Charlie. And I'm delighted to be here. You just have to excuse me. I should be asleep at the moment. So today what I'm going to talk to you about is GABA, and I'll have a bit of an excursion into glutamate as well, because we all sort of think we understand what glutamate's doing, so we'll sort of skate over the top of that and then talk about excitation and inhibition, because glutamate is the major excitatory neurotransmitter in the brain, and as we know, GABA is the major inhibitory one, so you have to think about the two of them together. Then I'll talk about the energy budget of the brain, so how much glucose and oxygen it has to burn in order to do its various tasks, which of course is an estimation, because it's continually being revised every time people do new experiments, but it's relevant here. So we'll have glutamate and the BOLD response, and then we'll talk about inhibition and the energy budget. So how expensive is inhibition compared to excitation? And then there'll be a bit of neurochemistry, and then we'll talk about the power of GABA, because it is actually a very powerful molecule. So glutamate and GABA, are they the yin and the yang of brain metabolism? So they're very similar molecules. You've seen the structures before. So they're connected just by a single enzymic reaction, glutamate decarboxylase, which basically takes that little red molecule that you can see, the carboxylic acid, off. It's decarboxylated, you lose it as carbon dioxide, and you end up with GABA. So I have a poll now, so be interesting to see what happens. The question for you, it says this slide will display live content. Okay. Well, the question for you was actually, what is the proportion of excitatory to inhibitory synapses? And this should be loading the poll, and it isn't. So the question for you, which I'll just see if I can remember what the options were. There was one, is it 50-50? Is it 90-10? Is it 97-3? Oh dear. Is it 60-40? Any idea? Who votes for 50-50? Who votes for 90-10? 90% excitatory, 10% inhibitory? Who votes for 97-3? Who doesn't care? Okay. See if I can move on. The answer, which would have come up if everyone had voted, is actually 90 to 10%. So about 90% of the synapses in the brain are excitatory and about 10% are inhibitory. So if we look at the brain energy budget, so this is taken from David Atwell's work, and David's very good because every now and then he does these little calculations about what the total brain energy budget is. So if you're using up a certain amount of oxygen and glucose every day, what proportion of it is used for housekeeping reactions, what proportion of it is used for synapses, how much does the grey matter use compared to the white matter. And he's basically come up with this idea that the resting potentials and housekeeping using up a certain amount of the budget. So in white matter, it's a lot more as a percentage because the white matter isn't doing as much in terms of synaptic activity. In the grey matter, which is the one here on the left, he's got the housekeeping activities, which is things like lipid synthesis and moving your actin around and reaching out to make a synapse with somebody, as taking up about 25% of total glucose use. Resting potentials and action potentials add around, that's these APs and RPs, and the synaptic activity at about 43%. So that's all predicated on it mostly being glutamatergic activity. So it's quite expensive to run. It has been known for some time that there is coupling of brain glucose metabolism and glutamatergic neuronal activity. So Nikki Simpson showed this quite nicely in a paper from some time ago, in 1998. And this has been repeatedly shown by various people that there is this relationship between glutamate and glucose use and glutamatergic neuronal activity. So more excitation means more activity. Duh. Okay. The glutamate levels and the BOLD response tend to couple quite nicely. You can see here you've got an on-off stimulation, which fMRI people are familiar with, and your glutamate signal tends to track your BOLD response actually quite nicely. It is to a degree area dependent, and it is to a degree dependent on what your stimulus is. So you need to bear those things in mind. Of course, you know, in neurochemistry, nothing's simple. There's always complications. It could be this, it could be that. So you have to take these things into account. But generally, the two things correlate quite nicely with one another. So, you know, you'd actually expect that because this is the neurochemistry. We've got our Krebs cycle here. So isocitrate coming around to alpha-ketoglutarate and going off to bisuccinyl-CoA. So you can just imagine the Krebs cycle sweeping around here. And glutamate is in direct exchange, really, with alpha-ketoglutarate, which is one of the main control points in the Krebs cycle. It's also, the other thing that we often forget is these aminotransferase reactions, which convert glutamate to alpha-ketoglutarate, and in the process we'll add an amine onto things like oxaloacetate or onto pyruvate, and you end up with various amino acids like aspartate, which is also an excitatory molecule, and alanine, for example. It also directly makes glutamine, but these two reactions are cell-specific, and the synthetase actually requires ATP. So you're not in glutamine and glutamate are not necessarily directly connected to one another, whereas glutamate and alpha-ketoglutarate do tend to be, because these reactions here, particularly the aminotransferase ones, are fairly near equilibrium enzymes. So this means that if your TCA cycles, by the way, don't necessarily go faster, they tend to get fatter. So it's like a donut getting fatter. You put more molecules into the TCA cycle, which is how you get more flux through it. It does speed up, but it also gets fatter. But as it gets fatter, your alpha-ketoglutarate concentration increases and your glutamate concentration increases concomitantly because these reactions are in fairly fast exchange. So the bottom line for that is that glutamate and the TCA cycle are fairly nicely linked, and therefore brain energy metabolism and glutamate are fairly nicely linked. But what about GABA? People obviously think that GABA is important. If you look at the commercial activity around GABA at the moment, there's lots and lots of people busy flogging it off. This idea that you could eat it and it would end up in your brain seems a bit strange because it doesn't actually cross the blood-brain barrier. So if you're ever interested in buying these things, just remember that it's probably all just going down the toilet, not actually into your brain in the first place. But it's a big market. So what is GABA actually doing? So we've got a little bit of useful background facts here. Like I said, there's about 90% of cells and synapses are glutamatergic. So the other thing you want to consider around energy budgets is that postsynaptically, chloride ions, which is what you move when you have an inhibitory synapse, they move down a smaller electrochemical gradient at inhibitory synapses than sodium ions do at excitatory synapses, which tells you that there's probably less energy expenditure involved in hyperpolarizing as compared to polarizing a membrane. So there's various different types of inhibition, and this is important when you're considering what the GABA is actually doing. So you have presynaptic inhibition, which is what happens at the presynaptic terminus of a synapse. You also have postsynaptic inhibition, which is at the postsynaptic terminus. You have phasic inhibition, which is basically the sort of normal spiking GABA that is very short-lived. And you have tonic inhibition. And tonic inhibition is where you get sort of tone that goes on for some time and helps to control your entire network. So I'm going to talk a bit about the various types of inhibition because they're important when you're considering what your GABA is actually doing. Now I have another slide which probably isn't going to work. So the question was, what is the estimated relative energy cost of inhibitory synaptic activity? So we're talking now probably mostly about the phasic inhibition. There are the options, 10, 16, 3 and 0.5%. So remember that we said that the in grey matter housekeeping reactions were taking up about 25% of activity. How much do you think inhibitory synaptic activity is actually contributing? Well, this is good because the answer is actually 3%. And eventually we'll move on to the next slide. Thank you. So 3%. If it's this small, why do we need to bother about it? If it only accounts for 3% of brain energy, then as scientists, what's it matter? Well, because 3% of the brain activity is the synaptic activity, but there are a number of ways that GABA can actually significantly impact what's going on in the brain, and the rest of my talk really is going to be taken up with telling you about those. So the answer is yes. Well, I would say that, wouldn't I? Okay, the devil is in the details. We have a couple of metabolic pathways here, which we need to have a look at. So what we have here is the presynaptic neuron that I mentioned, the postsynaptic neuron, and an astrocyte. There's also oligodendrocytes in the brain, which actually are now emerging to have quite strong metabolic activity, but all the models actually ignore them. So just be aware of that. So GABA, as I said, is produced in the presynaptic neuron, for example. It has to be a GABAergic neuron because if it's not a GABAergic neuron, it doesn't have the enzyme glutamate decarboxylase that actually makes GABA. So it's made in GABAergic neurons. It's produced from glutamate, and it can be incorporated into a vesicle where it can be released at a synapse as GABA. It can then has a number of different fates. It could go and bind to a particular type of GABA receptors, the GABA-A receptor. Now, the GABA-A receptor, when I say the GABA-A receptor, there's actually multiple different types of GABA-A receptors. It's made up of alpha, beta, gamma, delta, epsilon, pi, and rho subunits, which there's multiple different versions of each of them, like there's 6 alpha subunits, for example. Luckily, you have to have an alpha, a beta, and a gamma subunit, and then you could possibly include one of the others. And that sort of narrows down the number of possible GABA-A receptors we could have to around 10,000 different types. In practice, there's probably only about 13 to 15 different types of GABA-A receptors expressed in significant quantities in the brain. But just be aware that all GABA-A receptors are not equal and they all do different things. So you have GABA-A receptors which live in the postsynaptic neuron, but you also have GABA-A receptors which live out here, which is what we call perisynaptic or extrasynaptic receptors, and they play very interesting and specific roles in inhibition. We also have the GABA-B receptor, which luckily only has 2 subunits. There's the B1 and B2. Sounds like the bananas in pyjamas, but and then you have a couple of different splice variants of those. And those are metabotropic receptors. So instead of letting ions in and out, they're coupled to a G protein, which then affects the local environment, so calcium channels or chloride channels. The other thing we have here is these GATs, which are GABA transporters. So they're responsible for removing the GABA from the extrasynaptic space or from the extracellular space once it's been released. So if you have release of neurotransmitter, then your transporters will actively take up GABA. And the interesting thing about these transporters is that they can actually work in reverse. So they can release GABA. And they do that depending on what the local chloride potential is. So they're activity dependent. So if you've got activity, you can have release of GABA. So just bearing all those things in mind, we also have a way of metabolizing GABA once it's been made through GABA transaminase, which is an enzyme that basically reincorporates GABA back into the TCA cycle at the succinate level. And that'll become interesting in a minute as well. Now this looks awful, but I'm not going to go through this. Everyone says. Astrocytes can actually also make GABA. They don't make much, but you need to be aware of it. So it's made through this cute little pathway over here, putrescine, which sounds horrible. It's actually a type of biological amine, and it's active at NMDA receptors. But ornithine, if you add ornithine to brains, you actually end up with quite a lot more glutamate and GABA, either through this pathway, which is the main one, or through the metabolism of putrescine, which is the bit that can occur in astrocytes. And that links GABA metabolism through to creatine metabolism, arginine, and nitric oxide. So that's another little pathway that people just usually ignore because it's complicated and it's not really known what activity, what the impact of it is. So back to this pre- and postsynaptic activity at GABA B receptors. So the presynaptic activity can change the amount of calcium that you've got, and the calcium is important for basically release of neurotransmitter. So if you have GABA binding to the GABA B receptor, it changes the calcium levels and basically means that the presynaptic neuron releases less neurotransmitter. So this is a way that GABA can interact with the glutamatergic system or with the release of neurotransmitter. So these GABA B receptors could also be on non-GABAergic neurons. The postsynaptic one is linked to potassium channels, and when it activates the potassium channel, it means that the action potential that you get through the postsynaptic neuron is actually can be lowered. So it has a way of sort of turning off specifically the postsynaptic neuron. So what does this actually mean in terms of the energy budget? So this is the calcium channel, and this is the chloride channel, and it has a negative effect. So in terms of the energy budget, so this is a hypothetical. So if we say resting potential, 80% of our activity is excitatory and 20 is inhibitory. We actually know it's more like 90-10, but for the sake of this, we'll just pretend it's 80-20 because the numbers are easier to deal with. So if we apply an agonist at GABA B, if the agonist has only a slight effect, what it tends to do is basically decrease the excitatory but increase the inhibitory response. Now, inhibition is not an energy neutral or an energy negative thing, because to inhibit a neuron you have to hyperpolarize it, and that means you have to be pumping sodium and potassium. So there is an energy cost associated with it. The resting potential is it's called resting potential for a good reason, because it's the most energy efficient place for the neuron to be. So if you hyperpolarize, you have to pump ions, and that takes a bit of energy. So in this case, we've turned a bit of excitatory down, but we've got more activity at the inhibitory neuron because of our hyperpolarizing. And in fact, that lends to a slight increase in the overall energy budget. So we've got basically an increase with the agonist. If we turn the inhibition up slightly more, we get a bigger reduction in excitatory activity for pretty much the same energy budget, and you get an overall decrease in metabolism. So an agonist at GABA B could give you an increase in energy demand, or it could give you a decrease in energy demand. Ditto with an antagonist. At this point, everyone has to stand on their head to understand what an antagonist is doing. So it's actually switching off the off switch. So your agonist is actually basically increasing your excitation with a decrease in inhibitory activity. And depending on how much you put in, it also can give you more or less energy demand. So an agonist or activity at GABA A receptors, just because you've turned the receptor on, doesn't necessarily mean you've got less total energy demand. It's a bit complicated, but that's how it works. So what about tonic inhibition? Well, that's the really interesting thing for us using MRS to measure GABA. So with tonic inhibition, what we're talking about here is extrasynaptic release of GABA by these transporters, and there's also there's various bits of evidence in the literature supporting extrasynaptic release of GABA. So once it's released, it can bind to either these extrasynaptic GABA A receptors, and they tend to be more of a master switch. And to show you about that, I'll show you some results from an experiment that we did a while ago. So we used a compound called vigabatrin, which inhibits GABA transaminase. So it stops GABA being recycled back into the TCA cycle. And what that does is it increases extrasynaptic concentrations of GABA. So not the GABA that's sitting there in the vesicles, but the GABA that's actually hanging around being metabolic GABA in the extrasynaptic space. So put vigabatrin in and your GABA concentrations go up because it's a nasty inhibitor. It kills the enzyme, basically. So these are experiments with brain slices, and you don't need to know any of the details of this apart from up is excitatory or more metabolism, and down bars are less metabolism. So if we put AMPA, which activates the glutamate AMPA receptor, into brain slices, basically we get an awful lot of metabolism. If we put this thing in, which is AXIT metabotropic glutamate receptors, we get a big increase in metabolism. If we put this in, which is a GABA B antagonist, we get a mild increase in some bits of metabolism, but not in others. If we put these drugs in along with vigabatrin, so vigabatrin's upped our GABA levels, and we've put these activators in, and we get a big decrease, and we get exactly the same big decrease with all of these. So no matter how you activate the slices, as soon as you have more GABA, once the slices are activated, you get a big turn-off of everything that's going on. But the interesting experiment is this one, which is where we added in this compound called TPMPA, which is an antagonist at GABA A receptors and also at GABA C receptors, which are actually a type of GABA A receptor that has Rho subunits only. And they're very, very sensitive extrasynaptic receptors, so they respond to very low concentrations of GABA, but they're not in the synapse. So it's where you get this overspill of the GABA coming out of the synapse. So you've got too much activity, you have all this GABA being released, and it activates these receptors. And when you put TPMPA in, it has normally got a very strong excitatory response, but when you put it in with the vigabatrin, they cancel each other out, which tells us that wherever TPMPA is acting, and unfortunately, like most GABA drugs, it's not clean, it acts at more than one receptor. So we don't quite know what the culprit is, but we know it's got to be some sort of GABA receptor. We get this very nice, almost nothing happening activity. So that shows you how powerful GABA can be. So you've gone from really, really active to everything switched off, just with a tiny little bit of extra GABA. And this has been shown to be cell-specific and act through various receptors. Most people are pinging the alpha-6 containing GABA A receptor, but yet again it still hasn't been nailed down. GABA can also directly modulate blood flow, so there's quite a bit of evidence now that it directly affects the vasoconstriction. It may do this itself through GABA A receptors, or it may do this through a neuropeptide signalling system like neuropeptide Y. And I've just put a few papers up here. There's a plethora of them in the literature about how GABA can impact cerebral blood flow. So, what do bulk GABA levels actually mean? It's a good question, and one that we're going to have to keep asking, but it's brain region dependent. So depending on, remember, every time you place a voxel in the brain, that voxel is going to have a composition of multiple different types of cells. So MR people see things in black and white. So we've got grey matter and we've got white matter, and we get really obsessed with parcellating our voxel. That voxel also contains multiple different types of neurons and oligodendrocytes, which people ignore, and astrocytes, and different populations of GABA-containing cells. So it is very brain region dependent what happens with GABA. It's also activity dependent. As I showed you, if you activate the slices, you can actually then have this feedback mechanism whereby the GABA is released and everything gets switched off. And I think my colleague over there will be discussing this. You cannot distinguish neurotransmitter and metabolic pools. Every time I read a paper where people say, I've measured neurotransmitter GABA, yeah, you can't. The two things are certainly not in a 27 cubic centimetre, centimetre cube box, which is what you're measuring with megapress. The neurotransmitter and the metabolite are in fast exchange. So it can be a neurotransmitter 1 minute and it's back to being metabolic GABA the next. So you can't distinguish between the two, and you've got no way of knowing what the proportion of each is because it's a bulk measurement. it does seem that total GABA levels are related to this tonic inhibition, and the more GABA you have, the more likely you are to be able to turn things off in a hurry. If you have low levels of GABA, the amount of GABA that you've got available to release through the GATs, through the transporters, is relatively low. But of course, the research is ongoing. We're finding out new things about the GABAergic system all the time, and every time people publish papers having measured GABA in vivo, it adds to that literature. So go and do it. Go and measure it. And I thank you for your attention.